A human skeleton buried 10,000 years from now may still look unmistakably human. Yet the most important changes may be hidden in places that bones cannot fully reveal: in growth, immunity, fertility, metabolism, and the way a body is kept alive. The last 10,000 years changed us through farming, settlement, disease, and medicine. The next 10,000 will begin in conditions no earlier human population ever faced. For the first time, technology may shape the body as strongly as climate, food, or physical work once did. The future human may not look dramatically different at first glance. The deeper changes may be harder to see and far more important. What would 10,000 more years do to the human body? The piece of evidence is smaller than a grain of rice. It may come from the dense bone around the inner ear, from the root of a tooth, or from another part of a skeleton where fragments of DNA survived after the rest of the body disappeared. Inside that damaged material, researchers sometimes find a single position on chromosome 2 that helps reveal whether an adult was likely to continue producing lactase, the enzyme that breaks down the milk sugar lactose. Nearly every healthy human infant begins life able to digest milk. In most people across the world, lactase production normally declines after childhood. Yet in several populations with long histories of dairying, genetic variants arose that can help keep the lactase gene active into adult life. One of the best studied European variants is known as RS49882235. It lies in a regulatory region within a neighboring gene called MCM6, rather than inside the lactase gene itself. A tiny change in a region that helps control another gene could alter what an adult intestine was able to do. This is one of the clearest reasons that 10,000 years cannot be dismissed as too short for meaningful human biological change. The ancient DNA record suggests that the European variant associated with lactase persistence remained uncommon for much of the period after farming and animal domestication began. Milk was already being used, yet many adults probably did not digest large amounts of fresh lactose in the way that many Northern Europeans can today. Archaeologists have found chemical traces of dairy fats in prehistoric pottery. A large study examined roughly 7,000 pottery residues from more than 550 archaeological sites, tracing milk use across about 9,000 years of European prehistory. The vessels show that people were exploiting milk long before the best-known persistence variant became common. Culture moved first. Bodies followed later, unevenly, across generations. That delay matters. It prevents the story from becoming a simple tale in which the first person to milk an animal immediately created a population able to drink fresh milk. Early herders had other choices. Milk could be fermented, soured, turned into curds, mixed with other foods, or consumed in amounts that did not overwhelm the intestine. Fermentation and cheese-making can reduce lactose. A community did not need widespread adult lactase persistence before it could make use of dairy animals. The pottery and the DNA therefore record two different histories. One is the history of a practice. The other is the history of a biological response to that practice. They overlap, though they do not follow the same line. This distinction is central to any attempt to look 10,000 years ahead. A new technology can spread rapidly through learning, imitation, trade, or necessity. A heritable biological response usually depends on births, deaths, family size, migration, chance, and many generations of different reproductive outcomes. The environment created by culture may remain in place for centuries, before a genetic response becomes visible. It may never produce one if the cultural solution works well enough on its own. In the case of milk, something eventually favored lactase persistence strongly in several populations. Ancient European genomes indicate that the best-known persistence allele was still at low frequency well into the Bronze Age, then rose greatly during the last few thousand years. The exact path remains under study. Researchers once told the story mainly as a direct nutritional advantage. Adults who could digest fresh milk gained more calories, fluid, protein, and minerals from their herds. That advantage is plausible, but the archaeological and genetic record has made the explanation less tidy. Milk use was widespread in many places where the persistence allele stayed rare for a long time. The intensity of dairying alone does not fully explain the later rise. Some researchers have proposed that the advantage became especially strong during famine or infectious disease. Undigested lactose can draw water into the intestine and be fermented by gut microbes, sometimes producing diarrhea and other symptoms. In an otherwise healthy person with access to different foods, this may be manageable. During severe food shortage, intestinal infection, or dehydration, the consequences could have been more serious. The evidence does not allow us to watch a single prehistoric crisis and declare that it caused the genetic change. It does suggest that the value of a trait can depend on rare, difficult periods rather than ordinary days. A variant may offer little advantage for years, then matter greatly when harvests fail, settlements become crowded, or disease moves through a population. This is how a body can be shaped by conditions that leave only partial traces in soil, bone, pottery, and DNA. The same gene can be almost irrelevant in one setting and valuable in another. The persistence variant did not make its carriers stronger in every sense, healthier in every environment, or biologically superior. It kept one digestive program active after childhood. Its usefulness depended on access to milk, the form in which that milk was consumed, the surrounding diet, the local burden of disease, and the history of the population carrying it. Even within Europe, present-day frequencies vary widely. Across much of East Asia, many indigenous American populations, and numerous other regions, adult lactase non-persistence remains common. That condition is not a failure or a recent loss. It is the ancestral human pattern, and many food traditions developed without relying on fresh milk in adulthood. The change also did not occur only once. Different genetic variants associated with adult milk digestion have been identified in parts of Africa and the Middle East. Several arose on different genetic backgrounds. This is a powerful detail because it shows that similar cultural conditions can lead to partly similar biological outcomes through different molecular routes. Evolution does not have to discover one universal answer and spread it everywhere. It can reach toward the same function more than once. Pastoral communities in East Africa did not simply wait for a European variant to arrive. Their own population histories contain other changes near the regulatory system controlling lactase. The broad pattern is called convergent evolution, though the details differ among populations and remain incomplete. What repeated was not one exact mutation across the whole species. What repeated was the pressure created when adults depended heavily enough on animal milk for long enough that continued lactase production could improve the chance of leaving descendants. This reveals another limit in the title's question. 10,000 more years will not do one thing to the human body everywhere. There is no single environment acting on every person, no single diet, no single disease landscape, and no single pattern of reproduction. Future populations may mix more widely, which can spread variants across regions. They may also become separated by economics, geography, technology, or even habitats that do not yet exist. A trait favored in one setting may remain neutral in another. A change that becomes common in one population may stay rare across most of humanity. The milk story gives us a real example of a large biological difference becoming common within the time span named by the video, while also warning us against turning that example into a prediction for everyone. It shows possibility, not destiny. There is a deeper lesson inside the position of the variant itself. The European persistence-associated change lies in a regulatory region. It does not build a new organ, it alters the timing of a familiar program. The infant intestine already produces lactase. The derived regulatory pattern helps prevent that activity from being reduced in the usual way after weaning. Evolution often works by adjusting when, where, or how strongly an existing gene is used. Over 10,000 years, a future human population would not need to acquire a new limb or a new organ for its biology to become meaningfully different. A small shift in gene regulation could alter growth, digestion, immune activity, fertility, or the response to a manufactured environment. These changes might be nearly invisible in a skeleton. They could matter every day inside living tissue. Yet the LCT region also warns against reading a single genetic marker too confidently. Recent large-scale analyses of ancient genomes have found a complex pattern of selection around the wider LCT region. Some signals in that region may have begun changing before the famous persistence allele itself rose. Genes sit near other genes. Variants travel together through ancestry, and migration can change frequencies without selection acting directly on every nearby position. Ancient DNA is powerful because it gives researchers samples from different times, rather than forcing them to reconstruct the whole past from living people. It is still incomplete. Preservation is uneven. Europe is sampled far more heavily than many other regions. Graves do not represent everyone who lived. A sequence from a skeleton can reveal a genotype, but it cannot tell us exactly how much milk that person drank, whether the person developed symptoms, or how many surviving children carried the same variant. The record becomes most useful when DNA is read beside pottery residues, animal bones, settlement patterns, isotope evidence, and the biological function of the gene. No single object carries the entire answer. Together, these traces show a culture changing its food supply, a digestive trait remaining rare for generations, then a marked rise in particular populations under pressures that researchers are still working to separate. This is enough to establish the first solid step toward the future. Human evolution did not end with the appearance of our species, with the spread of farming, or with the beginning of written history. A cultural invention created a new relationship between adults, animals, and food. That relationship changed the value of an inherited regulatory variant. Over many generations, the variant became much more common in some populations. 10,000 years was enough. The change was specific rather than total, regional rather than universal, and dependent on a world people had partly built themselves. The same principle is operating now, though the pressures are different and often harder to see. Processed diets, artificial light, antibiotics, reproductive medicine, pollution, climate control, global travel, and long periods of sitting are all cultural conditions. It does not follow that each will produce a genetic adaptation. Many may disappear too quickly, affect reproduction too weakly, or be managed by technology before inherited change can accumulate. Some may alter bodies during growth without changing DNA at all. Others may interact with existing variants in ways that only become visible after many generations. The milk example gives no list of future organs and no fixed portrait of a person living in the year 12,000. It gives something more reliable, proof that a behavior can change the environment, that the new environment can change which inherited traits are useful, and that human biology can respond within the span of 10 millennia. Still, genes are only the slowest layer of the body's response. Long before a useful variant rises through a population, bone, muscle, and connective tissue can change during a single childhood because of the loads placed upon them. The next evidence is larger than a DNA fragment. It can be held in two hands, measured along its shaft, and read in the thickness left by a life of movement. A cut through an ancient thigh bone can reveal how far a person's body was carried through the world. The outer wall of the femur is not a fixed shell formed once and left unchanged. During growth, bone is added, removed, and rearranged in response to the forces passing through it. Repeated walking over long distances, climbing uneven ground, carrying weight, and changing direction place bending and twisting loads through the shaft. Over years, those loads help shape the amount and distribution of bone around the hollow center. Researchers can scan a femur or tibia, reconstruct its cross-section, and estimate how strongly that shaft was built to resist particular kinds of bending. In one large European study, the sample included 1,842 individuals and extended from the Upper Paleolithic, roughly 33,000 years ago, into the 20th century. The clearest long-term change appeared in the lower limbs. Measures related to front-to-back bending strength in the femur and tibia began to decline during the Neolithic, around 7,000 to 4,000 calibrated years before the present, then continued downward through the Bronze Age and Iron or Roman periods. The upper limbs did not show the same directional pattern. That difference matters because a general decline, caused only by nutrition, illness, or smaller average body size, should not automatically single out the legs in the same way. The pattern is more consistent with a gradual reduction in habitual mobility, especially the repeated loading produced by moving across a landscape. It also refuses the simplest version of the farming story. Mobility did not collapse on the first day that seeds were planted. In the populations included in this study, the decline unfolded over thousands of years. As food production intensified, settlements became more permanent, and daily movement was reorganized. A femur does not preserve a diary of steps. It cannot tell us that one particular person walked 12 kilometers on a certain morning or carried a specific load home at dusk. Cross-sectional geometry is an indirect record shaped by body size, age, sex, development, inherited proportions, and activity. Researchers compare groups and long-term patterns because a single bone can be misleading. Even within one settlement, bodies may have experienced very different lives. Some people may have tended animals far from the houses. Others may have processed grain, carried water, worked fields close to home, traveled between communities, or spent long periods performing tasks that loaded the arms more than the legs. The remains that have been studied, therefore, show averages and ranges, rather than one universal Neolithic body. Yet the broad European pattern is difficult to ignore. The lower limb shafts change more strongly than the upper limbs. The change began near the adoption of food production, and it continued gradually as later economies became more settled and mechanically organized. The bone was recording a change in the geography of ordinary life. A foraging group may return to familiar places, remain within a known territory, or spend part of the year in a favoured camp. Farming does not automatically mean that nobody travels. The difference lies in how often the body must cross large areas, how predictable the routes become, how labour is divided, and how much daily survival depends on movement between scattered resources. Grain can be stored. Animals can be enclosed or guided. Water can be carried along repeated paths. Fields bring work into a smaller area, even when that work is physically demanding. A farmer may bend, lift, grind, dig, and carry for many hours, while still covering less varied ground than a mobile forager. The skeleton can preserve that distinction. Physical effort and mobility are related, though they are not identical. A body can work intensely without travelling far, and a body can travel far without repeatedly lifting heavy loads. This is why the lower limb evidence should not be reduced to a claim that early farmers simply became inactive. Many did not. Their work could be relentless. What changed was the pattern of loading, and bones respond to patterns. The shaft of a long bone is built like a tube because material placed farther from the center can improve resistance to bending. The direction of that material matters as well. A shaft, reinforced against front to back bending, carries a different structural history from one shaped more evenly around its circumference. Researchers use measurements such as second moments of area and section moduli to describe these properties, then adjust for body size before comparing individuals from different periods. An ancient leg bone is therefore more than a measure of thickness. It is a record of how force repeatedly entered the body while that body was still capable of adapting its structure. This capacity is strongest during growth. Childhood and adolescence matter because the skeleton is being constructed while activity is taking place. Adult bone continues to remodel, but the broad proportions of a long bone shaft reflect much of what happened before full maturity. Two people with similar inherited potential can develop different skeletal strength if their growing bodies experience different loads. This is a form of biological plasticity. It can alter the body within one lifetime without changing the DNA passed to the next generation. The distinction carries the story beyond the milk digestion variant. Lactase persistence showed that culture can change the value of an inherited trait over many generations. Long bones show that culture can also change the body before any genetic frequency has time to move. A child born into a village, a city, or a highly mechanized society enters a physical world built by earlier generations. Paths, floors, tools, transport, food systems, and work routines decide which forces will reach the skeleton. The child's bones answer those forces quietly as they grow. The answer may remain visible thousands of years after the tools and movements themselves have vanished. Inside the ends of the bones lies a second record. Beneath the hard outer surface, near the hip, knee, shoulder, wrist, and other joints, is a lattice of thin struts and plates called trabecular bone. This inner network helps distribute loads through the joint. A study comparing recent humans with fossil hominins used high-resolution imaging to examine trabecular bone in limb joints. The fossil sample included early Homo sapiens from before the Holocene, Neanderthals, and earlier hominins, alongside chimpanzees and recent modern humans. In that sample, recent humans stood apart through relatively low trabecular density across both upper and lower limb joints. The authors interpreted this as a late development in human evolutionary history, potentially linked to reduced activity and growing reliance on technological and cultural solutions. The result does not prove that every recent person has less dense internal bone than every ancient person. Fossil samples are limited, preservation is uneven, and different joints respond to different loading histories. It does show that the low-density pattern familiar in recent human skeletons was not simply present throughout the earlier hominin record examined in the study. The outer shaft and the inner lattice should not be treated as duplicate evidence. Cortical cross-sectional geometry tells us how the long tube of the bone was distributed to resist habitual forces. Trabecular architecture records the internal organization of the joint ends, where loads move from one bone to another. Both can respond to activity, though they do so on different scales and are measured in different ways. When both lines point toward lower habitual loading in recent populations, they strengthen the idea that a major transformation occurred in the mechanical environment of the human body. They also show why the word lighter can be misleading. A skeleton is not simply placed on a scale and found to weigh less. Researchers are observing changes in density, geometry, distribution, and inferred resistance to loading. A narrow shaft can still be well suited to the forces it regularly experiences. Bone is living tissue that balances strength, repair, mineral demands, and the energetic cost of maintaining material. The body does not preserve structures merely because they were useful to distant ancestors. It builds in response to the demands that arrive during life. A well-contextualized settlement can make this process easier to see. Çatalhöyük, on the Konya plain of central Anatolia, was occupied from about 7,100 to 5,950 BCE. Its tightly clustered mud brick houses, burials, animal remains, plant remains, and human skeletons preserve evidence from more than 1,000 years of early settled life. Bioarchaeological work there found that mobility patterns changed through the occupation, while population density, disease exposure, diet, and social conditions also shifted. The people of Çatalhöyük were not motionless. They farmed, herded, gathered resources, built houses, climbed onto roofs, carried materials, and moved through the surrounding landscape. Yet the organization of that movement changed as the settlement grew and subsistence intensified. This is the closer view missing from any simple contrast between wandering hunters and seated farmers. A permanent settlement can reduce some forms of travel while creating new forms of repetitive labor. It can load the spine, shoulders, knees, and hands in ways that a broad measure of distance traveled will not fully capture. The archaeological record also shows that transitions differed by region. Food production began at different times, relied on different crops and animals, and mixed with hunting or gathering for long periods. Studies from the central Mediterranean and other regions have emphasized this mosaic, showing that changes in body size, diet, activity, and health did not follow one identical sequence. The safe conclusion is therefore narrower and more useful. In several well-studied populations, especially across Holocene Europe, habitual lower limb loading declined gradually as food production and settled life expanded. Recent humans also show relatively low trabecular density across the limb joints, compared with the fossil hominins included in one broad comparative study. These patterns do not establish that agriculture made every human skeleton fragile. They show that the body's mechanical construction can shift when ordinary movement shifts, and that such change can become visible on an archaeological timescale. This matters for the next 10,000 years because the modern mechanical environment is changing faster than the Neolithic one did. Elevators, vehicles, machines, paved surfaces, remote work, and automated transport can remove loads from the skeleton before genes have changed at all. A child can grow within a world where very little force is required for food, movement, communication, or work. Another child may grow under heavy labor, long walking distances, repeated carrying, or organized physical training. Their bodies will still belong to the same species, yet their bones may be shaped by sharply different demands. No evidence allows us to declare that future humans will inevitably have thinner bones. Public health, architecture, exercise, occupation, nutrition, medicine, and new technologies can all redirect the result. Artificial gravity, powered clothing, or habitats beyond Earth could create mechanical conditions unlike any in the archaeological record. The reliable point is that the future skeleton will not wait passively for evolution. It will begin changing during every childhood according to the forces that culture allows to reach it. The first evidence came from a regulatory change near a digestive gene. The second comes from the walls and inner lattice of the skeleton. Together, they reveal two clocks running at once. One moves through inheritance across generations. The other moves through growth inside a single life. Both are controlled partly by environments that humans build. The next trace lies in the face, where the same principle becomes visible in a smaller space. When food is cut, ground, cooked, and softened before it reaches the mouth, the forces passing through the jaw during childhood begin to change, and the body answers again. A lower jaw from the Nile Valley can preserve two histories at once. One history belongs to the teeth, which began forming during childhood and kept much of their original size after they erupted. The other belongs to the bone holding them, which continued to respond to muscle, food, growth, and repeated strain. In Lower Nubia, between southern Egypt and northern Sudan, researchers examined 102 adults, represented by 69 crania and 97 mandibles across cultural horizons, extending from Mesolithic hunter-gatherers to later farming populations. The sequence covered roughly 11,000 years. The strongest difference appeared between the Mesolithic remains and the groups that followed. On average, the earlier hunter-gatherers had larger, more robust mandibles, while later farming groups showed changes in mandibular size and shape, consistent with reduced demands on the chewing system. The rest of the cranium did not follow exactly the same pattern. This made the mandible especially important because it seemed to record something more specific than ancestry alone, the changing work performed by the mouth. The investigators were careful about migration and population history, since new people can bring new skull shapes into a region. Even with that uncertainty, differences among early and later farmers continue to fit the idea that chewing behavior helped shape the lower jaw. The object itself is easy to picture. The mandible is a curved bone with a horizontal body carrying the lower teeth and two rising branches near the ears. The masseter closes the jaw from the cheek, while the temporalis descends from the side of the skull to the coronoid process. Each hard seed, strip of dried tissue, fibrous root, or poorly ground grain sends force through the teeth, the tooth sockets, and the body of the mandible. During childhood, these repeated forces become part of the environment in which the jaw grows. Bone cells remove tissue from some surfaces and add it to others. Muscle attachments pull on particular regions. The jaw does not simply enlarge in every direction when chewing becomes harder. Its overall form changes according to where force arrives and how often it returns. A study of living adults used computed tomography to compare the cross-sectional areas of the masseter and temporalis muscles with three-dimensional mandibular shape. The full sample included 382 people, while a smaller group of 62 reconstructed mandibles was used for detailed shape analysis. Larger chewing muscle cross-sections were associated with a wider, more trapezoidal ramus, a more substantial coronoid process, a more rectangular mandibular body, and a more curved basal arch. Simple measurements captured only part of the relationship. The whole shape carried the clearer signal. This does not allow an archaeologist to look at one jaw and calculate the exact toughness of every meal its owner ate. Muscle force is only one influence among age, sex, inherited proportions, tooth loss, illness, and population history. The association still explains how a cultural practice can become a physical structure. A child who must chew resistant food for long periods grows under one mechanical regime. A child whose meals have already been pounded, milled, boiled, fermented, or baked grows under another. The difference begins before either child can choose a diet. It is placed into the mouth by tools, cooking methods, stored foods, and the labor of other people. By the time the adult teeth have erupted, years of force have already helped set the dimensions of the bone around them. This is why food processing matters beyond nutrition. Grinding stones do part of the work that teeth once performed. Fire weakens plant tissues and connective fibers. Pounding breaks hard pieces before they enter the mouth. Pottery allows grains and legumes to be softened in water. Later mills produce finer particles, while industrial methods make many foods require only modest chewing. None of these changes guarantees one exact jaw shape, because human diets remain varied. Together, they reduce the average mechanical challenge in many settled populations. Global comparisons of human mandibles have found that subsistence strategy is associated with mandibular shape, with hunter-gatherer groups tending to differ from agricultural groups even after the strong effects of geography and shared ancestry are considered. The skull as a whole often reflects population history more clearly, while the mandible appears unusually responsive to the functional world of eating. The teeth complicate the story because they do not respond to chewing in the same manner as the surrounding bone. Enamel cannot remodel after it forms. The crown of a permanent tooth reaches its basic dimensions before eruption. Tooth roots can undergo limited change, and teeth wear, drift, or move within the jaw. Yet the tooth itself cannot simply become narrower during childhood, because the meal has become softer. The bony dental arch is more developmentally flexible. This creates the possibility of a mismatch. If teeth retain dimensions established through inheritance and early development, while the upper and lower jaws grow within a reduced mechanical environment, the available space can become smaller relative to the total width of the tooth row. Teeth may rotate, overlap, erupt outside the ideal line, or remain partly trapped. Dental crowding is usually defined through the difference between the space required by the teeth and the space available in the arch. Both sides of that relationship vary. Large teeth can crowd within an average arch, and an unusually short arch can crowd teeth of ordinary size. The archaeological importance lies in the different biological behavior of the tissues. Bone can alter its growth in response to function, while dental crown dimensions are far less responsive after formation. A study comparing mandibular and dental dimensions across the Near East, Anatolia, and Europe found that the transition to agriculture disturbed the way similarities in jaw form and tooth dimensions tracked one another. The authors argued that phenotypic plasticity in the jaw could produce faster change than the more genetically stable dimensions of the teeth. The mismatch should not be turned into a myth that prehistoric humans always possessed perfect teeth. One early anatomically modern human from Caesarean cave in Israel, often dated to around 100,000 years ago, already displayed crowding and malocclusion. That specimen is valuable because it prevents a false absolute claim. Misaligned teeth existed long before farming, refined flour, or modern orthodontics. Genetic variation, developmental disturbance, tooth size, jaw relationships, early tooth loss, and other factors can produce malocclusion in any period. What changed through later history was the frequency and distribution of certain forms in some populations rather than the first appearance of the condition. Tooth wear added another difference between older and more recent mouths. Many foraging diets carried mineral particles from soil, ash, stone grinding, or imperfect cleaning. Resistant foods also demanded prolonged chewing. The crowns wore down during life, sometimes dramatically. Wear could reduce the height and width of contacting surfaces, while continued eruption and movement of teeth helped maintain contact as material disappeared. This process did not create more bone, though it could alter how the tooth row occupied space over adulthood. In low-wear modern mouths, the crowns remain closer to their original dimensions for much longer. Third molars may attempt to erupt into an arch that has experienced little interproximal wear and limited forward movement. This is one reason wisdom teeth cannot be explained by jaw size alone. Eruption depends on growth timing, tooth angulation, local anatomy, genetics, and available space. The evidence supports a broad relationship between food processing, reduced chewing demands, jaw development, and some forms of crowding, while leaving room for substantial individual and population variation. The same caution applies to the phrase smaller jaws. Lower Nubian farmers did not simply become miniature versions of the Mesolithic people before them. Shape changed as well as size. In the Nubian sequence, later groups could have shorter and wider mandibular bodies, altered ramus orientation, or different combinations of facial dimensions. A jaw can become less robust without shrinking evenly. Farming also changed childhood nutrition, disease exposure, weaning, workload, and population movement. Each of these can influence growth. When a comparison finds a difference between hunter-gatherers and farmers, chewing is a plausible contributor rather than the sole cause of every millimeter. A large global analysis of skulls across the transition to agriculture reached a similarly restrained conclusion. Changes were consistent with softer diets in pre-industrial farming groups, especially in regions of the skull influenced by the major chewing muscles. Yet the measured dietary effects were relatively small compared with the deep imprint of migration, genetic drift, and population history. This makes the jaw a better guide to the future because it shows how visible change can emerge from several processes layered together. Part of the difference can arise within one childhood through plastic growth. Part can accumulate across generations if inherited variants affecting tooth size, facial growth, or muscle biology change in frequency. Part can arrive when populations move and mix. Part can be produced by dental care, extraction, orthodontics, surgery, and changes in the timing of treatment. 10,000 years from now, a skeleton may carry the combined record of all four. The future mouth will also grow in an environment more engineered than any early farmer experienced. Food is increasingly cut, blended, extruded, pressure cooked, purified, and designed for predictable texture. Infants and children in many societies receive soft foods for long periods. though the pattern varies greatly by household and region. At the same time, orthodontic treatment can expand arches, guide eruption, move teeth, or remove selected teeth. Surgery can reposition jaws. Dental implants can replace missing roots. These interventions change individual bodies directly, yet most do not alter inherited DNA. Their evolutionary effect would depend on whether the traits requiring treatment influence reproduction and whether treatment changes that influence across many generations. There is no sound basis for declaring that technology will make human jaws disappear or that wisdom teeth will inevitably vanish. Evolution has no plan to remove structures merely because they cause inconvenience. A variant related to missing third molars could become more common only under the right pattern of inheritance and reproduction. Cultural treatment may even reduce selection by allowing many different dental arrangements to function successfully. The safest forecast is therefore less dramatic and more revealing. If future children continue to grow with lower chewing demands, developmental changes in jaw form and dental arch dimensions could become common within each generation, even without a matching genetic transformation. The mismatch between teeth and available space could persist, worsen in some settings, or be redirected by preventive practices and clinical treatment. Different populations may follow different paths because diet, childhood behavior, healthcare, and ancestry will remain uneven. The face seen from the outside might change subtly, while the more consequential changes appear in eruption patterns, bite relationships, airway dimensions, or the need for dental intervention. Evidence is not strong enough to assign one destination. It is strong enough to show that the mouth remains responsive to a cultural environment. The first three traces now form a continuous line. A regulatory variant near the lactase gene showed that a new food practice could alter genetic selection over thousands of years. The shafts and joints of the limbs showed that built environments could reshape tissue during one life. The mandible shows both clocks meeting in one small region because inherited teeth and developmentally responsive bone can fall out of step. This is the final movement before the central turn. Farming did more than soften food. Permanent settlements gathered people, stored calories, concentrated waste, brought humans near domesticated animals, and changed the diseases moving between bodies. Once many bodies began living close together, the strongest pressures were no longer confined to muscles, bones, and chewing. They entered the immune system, where the costs of settlement could be carried invisibly from one generation to the next. A tooth taken from a grave can contain two genomes. Most of its surviving DNA may belong to the person whose jaw once held it. A far smaller fraction may belong to a bacterium or virus that entered the bloodstream before death and remained protected inside the tooth for thousands of years. The pathogen may have left no visible mark on the skeleton. There may be no wound, no damaged joint, and no written name for the illness. Yet short molecular fragments can still carry the pattern of ancient DNA damage, allowing researchers to distinguish them from modern contamination. A recent large survey screened genetic data from 1,313 ancient individuals spread across Eurasia and covering about 37,000 years. Researchers authenticated thousands of microbial detections, including traces assigned to more than 200 known human pathogen species. The sample was uneven, with most individuals coming from Western Eurasia, and burial populations cannot represent everyone who lived. Even so, the result reveals a world that bones alone had hidden. Disease was moving through human bodies long before physicians could describe it. and the number and kinds of infections visible in the record changed as human communities became larger, more connected, and more dependent on permanent places. The transition did not begin with one sudden epidemic. Early farming villages brought people close to stored grain, waste, standing water, domesticated animals, and one another. But the old claim that agriculture immediately released every major crowd disease is too simple. Small Neolithic communities often remained below the population size required to maintain infections that spread rapidly, disappear after exhausting susceptible hosts, then need a new supply of people to return. Settlements were also separated by distance, and many early farmers still moved animals, exchanged partners, visited seasonal places, or combined cultivation with hunting and gathering. Ancient pathogen surveys have so far found only a limited range of clearly authenticated infections in many early Neolithic populations. A study of HLA variation in 125 ancient individuals from farming communities in present-day Germany found no evidence that pathogen pressure rose dramatically from the early to the late Neolithic within that sample. The authors concluded that the heavier increase may have arrived later, particularly from the Bronze Age onward. This slower beginning matters because it places the pressure where it belongs. The new disease environment was built over generations. A few houses became a village. Villages became connected landscapes. Herds grew larger. Trade routes carried animals, food, tools, and people farther than before. Waste accumulated near wells and living areas. Harvest failures pushed communities towards stored foods with their own insects, molds, and rodents. Mothers, children, older adults, and people weakened by poor nutrition spent more time near others carrying infections. None of these conditions guaranteed a new epidemic, but together they changed the opportunities available to microbes. The environment was no longer only a forest, grassland, coastline, or river valley. It also included walls, roofs, animal pens, storage pits, paths between settlements, and the bodies gathered within them. One of the clearest examples is hepatitis B virus. Ancient viral genomes show that HBV was already infecting people in prehistoric Eurasia, including hunter-gatherers and Neolithic farmers. The ancient strains were not always the same as those circulating today. Some belonged to lineages that later disappeared or were replaced. Their presence shows that a long-lasting virus did not need a classical city before becoming part of human life. HBV can persist within an individual for years, particularly when infection begins early in life, allowing it to move through families and communities without producing the brief, obvious wave expected from an acute epidemic. In the German Neolithic HLA study, different immune haplotypes associated in modern populations with protection against hepatitis B changed in frequency, yet the broader protective function may have been maintained through different combinations. The interpretation remains cautious because present-day disease associations cannot be transferred perfectly into ancient environments. Still, the paired evidence is unusually close. Viral DNA demonstrates that the pathogen was present, while human DNA preserves variation in the immune machinery that encountered it. The machinery is concentrated partly within a region on chromosome 6 called the human leukocyte antigen system, or HLA. HLA molecules display small pieces of proteins to immune cells. They help the body distinguish its own tissues from cells carrying unfamiliar molecular material. There is no single best HLA type for every infection. A version that presents part of one virus effectively may perform differently against another pathogen, Diversity can therefore be valuable at the population level. When many HLA variants are present, a new pathogen is less likely to escape recognition in everyone. The same variation also carries costs. HLA types associated with effective responses to infections may be linked to inflammation, tissue damage, or autoimmune disease under other conditions. The immune system must react strongly enough to control a threat without continuing to attack after the threat has passed. Natural selection does not solve that balance permanently. It shifts the balance toward whatever protected reproduction under a particular history of exposure. Ancient genomes allow researchers to observe parts of that history rather than infer it only from living populations. In an analysis of more than 1,600 ancient Eurasian genomes, researchers detected a broad selection signal spanning roughly 8 million bases across the HLA region. The genetic trajectories did not point to one clean sweep produced by one disease. They suggested several changes beginning at different times and moving with different intensities. One variant between HLA-A and HLA-W began decreasing around 8,000 years ago. Modern association data connect that version with slightly different risks for intestinal infection and chickenpox, though those present-day associations do not identify the ancient pathogen responsible for the shift. Another signal within the region began rising roughly 4,000 years ago and increased more rapidly much later. The safest conclusion is that immune selection intensified and changed repeatedly, while populations, diets, mobility, and pathogen exposures were also changing. The Bronze Age becomes important here, not because every earlier community was healthy, but because several conditions began overlapping across much larger areas. From roughly 5,000 years ago in many parts of Eurasia, expanding pastoral societies, wheeled transport, denser exchange networks, and major population movements connected communities that had once experienced more local disease worlds. Ancient pathogen DNA now places Yersinia pestis, the bacterium associated with plague, across parts of Eurasia during the late Neolithic and Bronze Age. Those early strains were not identical to the bacterium that produced the medieval Black Death. Many lacked the YMT gene that later helped the bacterium survive inside fleas and supported efficient flea-borne transmission. Some early infections may therefore have spread by different routes. 3 ancient genomes from Britain show that Yersinia pestis had reached the northwestern edge of Europe about 4,000 years ago, while genomes from the Femora region of Russia reveal strains circulating around 3,800 years before the present. The presence of the bacterium does not prove that every burial represents a vast epidemic. It proves that a dangerous zoonotic pathogen was moving across very large distances within the growing networks of prehistoric Eurasia. A tooth positive for Yersinia pestis records an infection in one person. A cemetery containing several related cases can reveal more. Genetic work on Neolithic farming families in Scandinavia detected repeated plague infections across multiple generations, suggesting that the pathogen could return or persist within a connected community rather than appearing as a single isolated accident. Even here, the record remains incomplete. Some infected people leave no recoverable pathogen DNA. Others may die from causes unrelated to the microbe detected in their remains. The cemetery represents those who are buried there, not the full living population. The importance of the evidence lies in the repeated contact between a pathogen, human families, and settled or semi-settled social networks. Once such contact recurred, inherited differences in immune response could begin affecting which lineages left more descendants. Selection would still be difficult to see in a single generation. A village could lose many people to infection, while leaving no clear shift in the gene pool if the deaths were distributed randomly or if survivors had no difference in later reproductive success. A variant could improve survival yet remain rare because it began in only one family. Migration could introduce immune variants that looked like selection when their frequency rose. Population replacement could remove a useful variant despite its biological advantage. This is why ancient human genomes must be read beside ancestry. During the Neolithic and Bronze Age, Europe and Western Asia experienced large migrations and repeated mixtures among hunter-gatherers, early farmers, and pastoral populations associated with the steppe. A higher frequency of an immune allele in a later population may result from local selection, incoming ancestry, or both. The large HLA analysis used ancestry-specific genetic trajectories partly to separate these possibilities. Even then, the researchers described several proposed drivers as likely rather than proven. The immune region is highly variable, densely packed, and associated with many functions. A strong statistical signal does not preserve the name of the pathogen that produced it. What it does preserve is the shape of a trade-off. One allele in the ancient Eurasian analysis was associated in modern datasets with reduced risk of intestinal infection, but increased risk of chickenpox in the opposite genetic state. Other immune variants are linked today to asthma, inflammatory bowel disease, ankylosing spondylitis, psoriasis, Behçet's disease, or multiple sclerosis. These associations do not mean that ancient farmers suffered the same modern diagnoses at the same rates. Modern disease categories arise within modern lifespans, environments, diets, and medical systems. They do show that a gene affecting immunity can carry more than one consequence. A molecular setting that helped the body react forcefully to a recurring pathogen may leave a tendency toward excessive inflammation when the original exposure changes. The advantage and the cost can remain joined because one gene influences several pathways, or because nearby variants are inherited together. Over time, medicine may encounter the second half of a bargain made under conditions that no longer exist. This is one reason the human body of the future cannot be predicted by listing useful traits and assuming they will increase. Stronger immunity is not a single setting that can be turned upward without limit. An immune system that reacts too slowly can allow infection to spread. One that reacts too broadly can damage the tissues it is meant to protect. A population facing repeated childhood infections may be shaped toward one balance, while a population living with vaccination, antibiotics, clean water, and low exposure to parasites may experience another. Global travel can carry microbes between continents within hours, yet public health can interrupt transmission before genetic selection has time to act. A pandemic may affect billions of people while producing little inherited change if mortality is low among those who would otherwise reproduce, if treatment removes differences in outcome, or if the responsible variants do not affect reproductive success. A quieter infection present across many generations could leave a larger genetic trace than a famous crisis lasting only a few years. The bones from earlier truk recorded force, the jaw recorded chewing. Immune selection is harder to picture because the relevant struggle occurred inside cells and blood. Ancient DNA gives it a physical anchor. A fragment from chromosome 6 can be placed beside a fragment from a viral or bacterial genome recovered from the same broad period. The match is rarely exact enough to say that one pathogen selected one allele. Across hundreds and then thousands of individuals, however, the pattern becomes visible. Pathogens changed in distribution. HLA frequencies changed. The strongest immune adaptations in parts of Europe appear to have accumulated mainly after the beginning of the Bronze Age, rather than arriving fully formed with the first farmers. A broad survey of ancient microbial DNA likewise found that several important pathogen groups became more common later in prehistory and historical time, supporting a gradual rise in infectious pressure rather than one immediate Neolithic explosion. The distance to the central turn is now very small. Farming had changed food. Settled life had changed movement. Processing had changed the jaw. Larger and more connected populations then changed the invisible organisms passing between bodies. Through all of this, survival remained a filter. A body unable to control a common infection could disappear before leaving descendants, while a protective immune response could become more common, even when it carried a later cost. The next change would be different in kind. Humans would begin preventing some bodies from disappearing at all. Clean water, vaccination, antibiotics, surgery, blood transfusion, assisted birth, and intensive care would not end disease or remove selection. They would alter the consequence of traits that had once been tested more directly by death. The pressure built inside the settlement was still biological, but the answer to that pressure was about to become increasingly cultural. Once medicine could keep a vulnerable body alive and help it reproduce, the old relationship between anatomy, survival, and inheritance would no longer operate in quite the same way. A human pelvis lifted from an old burial does not preserve the birth that once passed through it. The bones form the hard margins of the birth canal, yet the infant is gone, the soft tissues are gone, and the movements of labour leave no complete record. Even so, the pelvis carries a limit that has shaped every generation of our species. The opening must be wide enough for a baby to pass, while the same structure supports the trunk, anchors muscles used in upright movement, contains pelvic organs, and holds a floor of soft tissue beneath the abdomen. Human childbirth takes place inside this crowded arrangement. The infant's head is large relative to the canal. The canal changes shape from inlet to outlet, and the head commonly rotates while descending. There is no single perfect pelvic form. A global study of 348 female pelves found substantial variation in birth canal shape among individuals and populations, much of it structured by population history rather than one universal design. Other work has shown that pelvic shape co-varies with stature and head size in ways that may reduce some mismatches. These findings make the central evidence more precise. Difficult birth does not come from one narrow measurement shared by all women. It emerges when the size, shape, position, and movement of one infant meet the particular anatomy and physiology of one mother. For most of human history, a severe mismatch between fetus and birth canal could become a biological boundary. Labor might begin normally, continue for many hours, then fail to advance. The head could remain above a constricted part of the canal. A shoulder could fail to pass. The uterus could become exhausted or injured. Infection and bleeding could follow. Archaeological skeletons rarely let researchers identify one exact case of obstructed labor, because the crisis occurred mainly in soft tissue. The evidence is therefore built from living anatomy, clinical records, heritability studies, demographic history, and evolutionary models, rather than from one ancient pelvis carrying an unmistakable label. This is a place where restraint matters. We do not know how often feto-pelvic disproportion occurred in every prehistoric population. We do know that without effective intervention, severe obstruction can threaten both mother and infant. and that modern surgery can allow many such births to end with 2 survivors. The physical opening has not vanished. The consequence of reaching its limit has changed. The relationship between infant size and maternal anatomy is also shaped by advantages on both sides of the boundary. Larger newborn size is often associated with better survival under some conditions, while a pelvis cannot expand without cost. A broader bony canal may place different demands on locomotion, hip function, pelvic floor support, and the soft tissues that resist pressure from the abdominal organs and fetus. Computer models of the pelvic floor suggest that enlarging the opening can increase deformation and stress, unless other dimensions of the tissue change with it. Recent genetic work also indicates that pelvic traits are highly heritable and tied to several competing outcomes, including obstructed labor risk, pelvic floor disorders, walking pace, back pain, and hip osteoarthritis. The pelvis is therefore not a doorway that evolution simply failed to widen. It is a living compromise among functions that do not all improve in the same direction. This compromise helped inspire a model known as the cliff edge model of obstetric selection. The image is simple. Across much of the normal range, increasing fetal size can carry benefits, and a relatively narrower maternal pelvis may carry benefits of its own. Near the edge, however, a small additional mismatch can cause the chance of a successful unassisted birth to fall sharply. Natural selection can move 2 traits toward that boundary because the ordinary advantages accumulate gradually, while the severe cost appears abruptly only in a minority of births. The result can be a population resting surprisingly close to an anatomical limit. This is a mathematical explanation for why fetopelvic disproportion may remain present even under strong selection against fatal obstruction. It is not proof that every difficult labor follows this model, and it does not reduce childbirth to head width divided by pelvic width. Presentation, fetal position, uterine contractions, maternal soft tissue, previous births, nutrition, disease, medical decisions, and chance all matter. The point of the model becomes clearer when surgery enters the record. A Caesarean section can move the infant through an opening made in the abdominal wall and uterus rather than through the bony birth canal. The procedure has a long and complicated history, though its regular use with greatly improved maternal survival is recent on an evolutionary timescale. Once surgery allows a mother and infant to survive a mismatch that might previously have ended reproduction for one or both, inherited predispositions contributing to that mismatch can continue into the next generation. The original cliff edge study estimated that regular Caesarean use could have produced roughly a 10 to 20% increase in the rate of fetopelvic disproportion over several decades. That number came from a model built from published estimates, not from direct measurements, showing that human heads have already enlarged or female pelves have already narrowed by a known amount. The authors themselves framed the result as a prediction about rates of disproportion under altered selection. This distinction is the center of the entire story. A hospital does not rewrite the pelvis during one operation. The surgeon does not make the newborn's future children larger. What changes immediately is the outcome. A combination of maternal and fetal traits that reaches the old anatomical edge may now pass through birth without the same loss of life. If parts of that combination are heritable, descendants may carry them forward. Over many generations, the distribution of risk could shift. It may shift only slightly. It may differ among populations with different access to obstetric care, different fertility patterns, different nutrition, and different reasons for performing surgery. Caesarean delivery is used for many conditions unrelated to a true mismatch between fetal dimensions and the maternal canal. A rising surgical rate therefore cannot be read as a direct measurement of evolution. The operation creates a plausible change in selection. The size and direction of the long-term biological response remain uncertain. A second study tested one prediction using records across generations. Mothers who had been born by caesarean delivery were more likely, on average, to deliver their own first child by caesarean than mothers who had been born vaginally. The increase was especially relevant where the original caesarean had been connected to arrested labor or suspected disproportion. Shared anatomy, shared genes, maternal health, clinical practices, family environment, and decisions by medical staff can all contribute to that pattern. The data cannot isolate evolution from every social and medical influence. Still, the intergenerational association fits the broader possibility that predisposition to difficult labor can run through families and remain visible after medicine changes survival. The deepest turn is larger than caesarean birth. Surgery provides an unusually clear case because it crosses a physical boundary that can be measured. The same principle appears whenever culture changes whether a biological difference prevents survival or reproduction. Insulin can keep a person with type 1 diabetes alive. Antibiotics can carry a vulnerable immune system through an infection. Blood transfusion can replace what injury or childbirth removes. Corrective surgery can repair anatomy that once limited feeding, breathing, movement, or fertility. Assisted reproductive technologies can allow conception under conditions that previously prevented it. Each intervention has its own biology, inheritance, access, and timescale. None permits a general claim that medicine is making humanity weaker. Many treated conditions are not strongly heritable. Many arise from new mutations or environment, and many treatments improve health without meaningfully altering reproductive differences. The shared change is narrower. Survival is increasingly filtered through a cultural system capable of separating biological vulnerability from immediate reproductive failure. Natural selection continues inside that system. It does not require wilderness, physical hardship, or death in childhood. Any inherited difference connected to the number of descendants can contribute, even when almost everyone survives. Fertility timing, partner choice, contraception, migration, education, economics, and access to healthcare can influence genetic frequencies more strongly than some diseases do. Medicine can weaken one selective pressure while creating another. A treatment may allow a trait to persist, then a side effect may reduce fertility. A technology may equalize survival across genotypes, while social patterns cause some lineages to have more children. The phrase survival of the fittest becomes misleading when fitness is heard as strength. In evolutionary biology, fitness concerns contribution to later generations, and culture has become part of the environment that determines it. This changes the meaning of the next 10,000 years. The old picture would place the human body beneath climate, famine, pathogens, and physical labor, then ask which bodies endure. The newer picture must include operating rooms, vaccines, neonatal care, gene screening, prosthetics, hormone treatment, and technologies that may not yet exist. These systems do more than protect a finished adult. They act during conception, gestation, birth, infancy, and childhood, where small differences can influence an entire lifetime. They may preserve biological variation that earlier environments removed. They may also direct development toward new averages without altering inherited DNA. A premature infant supported through weeks of neonatal care may survive because machines temporarily perform functions the body cannot yet sustain. That child's adult body is neither an untouched product of natural selection nor an artificial object. It is a human body whose development passed through a cultural extension of physiology. The pelvis remains the most tangible marker of this change, because its boundary can still be held in the hands. The inlet, midplane, and outlet have different dimensions. The sacrum curves behind them. The pubic bones meet at the front. The infant head must enter in one orientation, rotate through changing spaces, then allow the shoulders to follow. Variation in pelvic form means that roots differ among women. Soft tissues can move, joints can shift slightly, and birth positions can alter available dimensions, yet the bony canal still imposes limits. Medicine has not removed those limits from anatomy. It has built another route around them. 10,000 years from now, regular intervention could contribute to a higher frequency of some combinations that raise the chance of obstructed labor. The evidence does not justify saying that this will certainly happen, that human heads will grow larger, or that future women will lose the ability to give birth vaginally. The cliff edge model concerns a statistical distribution, not a prophecy about every body. Pelvic and fetal dimensions are influenced by many genes as well as maternal nutrition, health, age, parity, and development. Medical practice can change. Preventive care could reduce excessive fetal growth. Surgical techniques may become safer or be replaced. Reproductive choices and gene technologies could alter inheritance in directions that no model based on recent decades can predict. The honest conclusion is that obstetric medicine has created a new evolutionary condition whose long-term outcome remains open. That condition marks the midpoint of the journey. Earlier cultural changes altered food, movement, chewing, crowding, and infection. Bodies responded because new environments rewarded some forms more than others. Here, culture begins changing what happens when a body reaches an old limit. A pelvis and infant that cannot complete birth through the canal may still produce two living people. The anatomical mismatch remains real, while its former consequence is interrupted. From this point forward, the human body is shaped by pressures that technology removes, pressures that technology creates, and forms of biological variation that technology allows to continue. The immediate result is not the end of evolution. It is a different field of evolution, one in which survival alone no longer tells us which traits will move into the future. A line of ink in a parish register can preserve an evolutionary event more clearly than a bone. On Île aux Coudres, a small island in the St. Lawrence River in Quebec, Catholic records followed births, marriages, and deaths across families born between 1799 and 1940. The pages contain no genes and no measurements of the body. They contain dates, the age at which a woman first gave birth, the number of children she had, which children survived long enough to form families of their own. When researchers reconstructed those pedigrees, the average age at first reproduction among women declined from about 26 years to about 22 across roughly 140 years. Women who began reproducing earlier tended to have more children over a lifetime, and age at first reproduction showed enough heritable variation for the authors to detect a change in estimated breeding values beyond what random genetic drift alone was expected to produce. Social conditions still explained much of the variation. The island's religion, household economy, marriage customs, nutrition, and limited geography all shaped when births occurred. The result, therefore, cannot be transferred to modern humanity as a universal trend toward younger motherhood. It does establish something quieter and more important. Within a medically and culturally organized human community, differences in reproduction can still produce measurable evolutionary change over only a few generations. The evidence changes the meaning of survival. A woman in this population did not need to escape a predator or withstand a famine better than her neighbors for natural selection to operate. She only needed to leave a different number of descendants for reasons connected, even partly, to inherited traits. Evolutionary fitness is measured through contribution to later generations. A person may live to an advanced age and leave no children. Another may die younger after raising several children who also reproduce. From the perspective of gene frequencies, the second life can have the larger effect. Medicine can reduce deaths from infection, injury, difficult birth, or inherited disease, yet it cannot make every person contribute exactly the same number of genes to the future. Once the sharp filter of early death becomes weaker, differences in fertility, reproductive timing, partnership, and childlessness can occupy more of the evolutionary field. This is the immediate consequence of the point reached in the previous evidence. Culture protected more bodies from old anatomical limits, while selection continued through the unequal paths by which those bodies formed the next generation. A second record begins in Framingham, Massachusetts, where the Framingham Heart Study started in 1948. Researchers followed families for cardiovascular health, repeatedly measuring traits such as height, weight, blood pressure, cholesterol, blood glucose, age at first birth, age at menopause, and lifetime reproductive success. An analysis of women from the first two generations asked whether measured traits were associated with the number of surviving children and whether the traits contained enough additive genetic variation to respond. The study predicted slow changes towards slightly shorter stature, greater weight, lower total cholesterol, lower systolic blood pressure, later menopause, and earlier first birth in the descendants of that particular sample. These were statistical predictions produced from observed relationships among correlated traits. They were not direct observations of a newly evolved body, and they did not describe every American population. A measured trait may appear to be favored only because it is correlated with another trait closer to the true cause. Height, weight, blood pressure, glucose, reproductive timing, and social circumstances are woven together. A change in education, contraception, employment, migration, or medical care can reverse the association before many generations have passed. The Framingham result remains valuable because it demonstrates how weak an ordinary contemporary selection can look. It may act through a difference of a fraction of a child on average rather than through mass death. The body also tends to be shaped by stabilizing selection, which favors a middle range rather than constant movement toward an extreme. Data from the UK Biobank were used to examine reproductive success in relation to measured traits and genetic variants among a large contemporary British cohort. For several characteristics, including height and waist circumference, the evidence was consistent with lower reproductive success at the extremes. Signals also appeared for earlier age at first birth in women and higher body mass index in men, though the authors emphasized that the observed effects could be indirect and were specific to the population and period sampled. A gene associated with body mass may influence appetite, puberty, health, behavior, or another trait connected to family size. A social environment can then magnify or suppress that relationship. Selection detected in one birth cohort may weaken in the next when education expands, contraception changes, housing costs rise, or the age structure of partnership shifts. The body of the future cannot be forecast by extending one short statistical arrow for 10,000 years. The arrow itself can turn. Modern genomic studies bring the evidence closer to DNA. In 2023, researchers combined data from 785,604 people of European ancestry and searched across the genome for variants associated with the number of children ever born or with having no children. They identified 43 independent genomic signals. The implicated biology extended across puberty timing, age at first birth, sex hormone regulation, endometriosis, menopause, and other reproductive processes. One coding signal near RGAP27 was associated with a higher number of children alongside a shorter reproductive lifespan, suggesting a trade-off between reproductive intensity and reproductive aging. Several associations were tiny, and the sample's ancestry limits how widely the results can be generalized. The number of children recorded in a biobank is also shaped by personal choices, social opportunity, partnership, contraception, infertility treatment, and historical circumstance. Even with those limits, the study found a physical bridge between reproductive success and inherited variation. Some genetic differences were being passed onward at slightly different rates within a modern population, living among hospitals, vaccines, supermarkets, and advanced reproductive medicine. One signal reached backward into the earlier story of food. The FADS1 and FADS2 region on chromosome 11 helps regulate the conversion of shorter dietary fatty acids into longer-chain polyunsaturated fatty acids used in cell membranes and many signaling processes. Variants in this region have shown evidence of selection over thousands of years, with histories that differ across populations and diets. The large reproductive success analysis found that an allele within the FADS region carried a signal of ongoing selection in the contemporary data as well. The direction should not be reduced to a claim that one modern diet is producing a better human metabolism. FADS variants can have several effects, and reproductive associations may travel through nutrition, hormone biology, ancestry, or linked variants. The value of the evidence lies in continuity. A region shaped during earlier dietary transitions may still be changing in frequency because its variants remain connected to reproductive outcomes today. The ancient environment did not vanish when medicine arrived. Older genetic trade-offs entered the modern hospital, household, and food system with us. Reproductive medicine adds another layer without giving evolution a simple direction. Contraception can separate sexual activity from birth. In vitro fertilization can allow conception when fertilization, sperm transport, ovulation, or other processes would otherwise prevent it. Donor eggs and donor sperm can separate social parenthood from genetic transmission. Embryo testing can reduce the chance of transmitting some severe single-gene conditions, while most common traits remain influenced by many variants and environment. Caesarean delivery can change the result of a difficult birth. Neonatal care can support an infant through a stage that once carried far greater mortality. Each technology alters a different link between genotype, body, survival, and reproduction. Their combined effect cannot be described as relaxed selection across the whole genome. Selection may weaken against one condition, remain unchanged against another, and intensify through a new reproductive pattern elsewhere. A treatment can preserve a variant that once reduced survival. The same treatment may be expensive, or unevenly available, producing differences in who benefits. A reproductive technology can increase the descendants of people with one form of infertility, while age at parenthood introduces other biological constraints. Culture does not stand outside evolution. It becomes part of the environment through which inherited differences meet real reproductive outcomes. This is also why longer life does not automatically create stronger selection for long life. Natural selection is strongest on traits that affect survival and reproduction before or during the reproductive years. A benefit arriving mainly after reproduction can have a weaker effect on the number of descendants, though grandparents and extended families may influence the survival and success of younger relatives. Modern adults often reproduce later, and many remain economically or socially involved with children for decades. That can extend the ages at which health affects family outcomes. The relationship still varies across societies. A disease appearing at 70 may matter little to direct reproduction in one population and influence years of care, wealth transfer, and support in another. Medicine can extend life far beyond the age of first reproduction, creating bodies that spend more years carrying genetic vulnerabilities whose effects earlier populations seldom survived long enough to experience. This changes disease patterns more quickly than it changes gene frequencies. It also creates new environments in which future selection may operate. Cancer risk, cardiovascular disease, dementia, and reproductive aging can share biological pathways with growth, repair, and fertility. A variant that supports reproduction early in life may carry a cost later, allowing both effects to remain together. The records from Quebec, Massachusetts, Britain, and the international biobanks do not point toward one future shape. They reveal the movement of selection away from a simple contest over immediate survival. Timing becomes important. The number of children matters. Childlessness matters. The interval between generations changes how quickly gene frequencies can move. Migration mixes variants between populations. Assortative mating, where people with partly similar traits or social backgrounds tend to form families together, can change how variants are combined. Economic conditions can influence family size more strongly than a small biological effect, then biology can contribute modestly within those conditions. The resulting process is real, though it is difficult to separate into a clean list of causes. Researchers must distinguish inherited influence from shared family environment, direct genetic effects from correlated traits, selection from migration, and genuine change from the unusual composition of a volunteer biobank. A large sample can make a very small association statistically visible without making it biologically powerful. Over 10,000 years, small differences can accumulate only when their direction remains reasonably stable. Human cultural environments rarely remain stable for long. The coming centuries may alter reproduction through longer education, urban living, declining or rising fertility, artificial gametes, gene editing, climate migration, or medical access. No current dataset can tell us which of these conditions will endure. The secure conclusion is limited to the present hinge. Natural selection did not stop when survival became more protected. It became less visible and more entangled with choices, institutions, and reproductive technology. The human body will continue to inherit variants at unequal rates, though the traits favored in one generation may lose their advantage in the next. This moves the story away from the midpoint. The old filters have been interrupted, yet no single new filter has replaced them. There will be no uniform pressure acting on every population. To look further into the next 10,000 years, the evidence must first abandon the idea of one future human body and follow the different environments in which future bodies may be formed. A tooth from a late Neolithic burial in the Basque Country can carry a future that never became universal. Researchers recovered ancient DNA from individuals buried there roughly 5,000 to 4,500 years before the present and examined a genetic variant associated with continued lactase production after childhood. In the individuals who could be studied, the estimated frequency of lactase persistence was about 27%, far below its frequency in many people living in the same broad region today. The sample was small, preservation was incomplete, and it cannot represent every community in southwestern Europe. Its importance lies elsewhere. By that period, people had already lived with domesticated cattle and dairy foods for thousands of years, yet the biological response remained uneven. Some adults likely continued producing substantial lactase. Many others probably followed the older human pattern in which lactase activity declines after weaning. The same cultural practice had not created the same digestive body in everyone. That unevenness has never disappeared. Adult milk digestion is common in several populations with long histories of dairying, though the responsible variants and their frequencies differ greatly across the world. In Europe, the best-known variant lies within a regulatory region near MCM6 and LCT on chromosome 2. In a study of 470 people from Tanzania, Kenya, and Sudan, researchers identified 3 other variants associated with lactase persistence. These variants appeared on genetic backgrounds different from the common European form and from one another. One showed evidence consistent with a strong rise during approximately the last 7,000 years. Similar digestive ability had emerged through several molecular routes because different pastoral populations placed milk into adult diets under their own histories of climate, disease, migration, and food scarcity. The shared result did not require a single mutation spreading across all humanity. This is the first clear reason there will probably be no single future human body. Evolution acts on inherited variation already present within a population, along with new mutations that arise by chance. The result depends on which people carry those variants, what environment they inhabit, whom they have children with, and whether the trait changes reproductive success in that particular setting. 2 populations can meet a similar problem through different biological routes. They can also meet the same problem while one population changes genetically and the other relies on food preparation, medicine, trade, or behavior. A dairy community can ferment milk and reduce its lactose. Another can depend more heavily on fresh milk during drought. A third can abandon dairying when its economy changes. The pressure may strengthen, weaken, or disappear before one genetic form becomes common. 10,000 years supplies many generations, yet it does not supply one direction. Ancient DNA from Western Eurasia makes this visible across a much larger scale. One major analysis used 1,664 reconstructed ancient genomes containing more than 8.5 million genetic variants. The remains extended from about 11,000 to 1,000 calibrated years before the present and came from a wide region stretching from the Atlantic coast toward Lake Baikal and from Scandinavia to the Middle East. Researchers separated parts of these genomes into ancestry paths connected with Western hunter-gatherers, Eastern hunter-gatherers, Caucasus hunter-gatherers, and Anatolian farmers. These were analytical categories representing broad population histories rather than four fixed human types. The groups moved, divided, met, and mixed repeatedly. Their genetic traces remain distributed unevenly in living people. Western hunter-gatherer related ancestry is relatively high in parts of the Baltic and Eastern Europe. Steppe-related ancestry reaches higher levels in portions of Northern Europe. Anatolian farmer-related ancestry is more concentrated around the Mediterranean and in parts of the Near East. No ancient ancestry is spread evenly across modern Eurasia. The same study found that natural selection also followed different ancestral paths. Near MCM6 and LCT, the famous European lactase persistence allele appears to have begun rising around 6,000 years ago. Other variants across the wider region showed signs of increasing earlier, suggesting that the selective history around adult milk digestion and energy use was more complicated than one mutation appearing after the first farmers began milking cattle. When the researchers separated genetic trajectories by ancestry, much of the signal came through Eastern hunter-gatherer and Caucasus hunter-gatherer backgrounds. Migration later brought these backgrounds into new combinations. A change observed in a later population could therefore reflect selection, incoming ancestry, or both. The distinction is essential. A trait can become more common because its carriers left more descendants within a community. It can also become more common because a different population arrived. The human body seen in any century is built from both processes. Another set of evidence lies in the FADS gene cluster on chromosome 11. FADS1 and FADS2 help regulate the conversion of shorter dietary fatty acids into longer-chain polyunsaturated fatty acids used in cell membranes and signaling. The importance of a particular variant depends partly on what the diet supplies directly. Diets rich in certain animal or marine foods can provide more long-chain fatty acids. Diets relying heavily on plants may place greater value on the body's ability to produce them from shorter precursors. Ancient and modern DNA suggests that selection in this region changed across both time and geography. One study found evidence that alternative FADS alleles were favored before and after farming began in Europe, with the later farming-associated signal stronger in southern Europe. The larger ancient genome analysis placed part of the selection associated with plant-heavy diets in Neolithic populations before some of those farmers entered Europe, followed by continued change as populations moved north and west. The body was responding to regional food systems rather than to an abstract event called agriculture occurring identically everywhere. This regional pattern could continue far into the future even if food becomes globally traded. Supermarkets can carry similar products across continents, while actual diets remain divided by income, culture, religion, local agriculture, public policy, allergies, medical advice, and access to fresh food. Some future populations may depend heavily on engineered plant proteins. Others may obtain fats and amino acids from cultured animal tissue, marine systems, insects, traditional herding, or food sources that do not yet exist. Technology can make one diet possible without making it universal. A metabolic variant that matters under one food system may have little effect under another. Medical supplements can weaken the disadvantage of a limited pathway. Scarcity can restore that disadvantage during war, migration, crop failure, or disrupted supply. Over 10,000 years, the direction of selection could change several times before leaving a stable trace. the immune system carries an even more divided record. Across an 8 million base region on chromosome 6 containing the HLA system, ancient genomes reveal several selection signals beginning at different times and moving in different directions. HLA molecules help present pieces of proteins to immune cells, influencing how the body recognizes infection. A variant useful against one pathogen can raise vulnerability to another condition. In the ancient Eurasian analysis, one allele began declining around 8,000 years ago and was associated in modern data with a lower risk of one intestinal infection pattern at the cost of a different risk involving chickenpox. Another began rising around 4,000 years ago and was associated with protection against some infections alongside an increased risk of psoriasis. Modern associations cannot identify the exact ancient disease responsible for each frequency change. They demonstrate the kind of bargain immune selection can leave behind. Protection in one environment may become inflammation in another. Population history made those bargains uneven. Ancient genome research suggests that several immune variants now associated with multiple sclerosis risk rose among pastoralist populations of the Pontic steppe and entered much of Europe with migrations related to the Yamnaya about 5,000 years ago. Researchers propose that these variants may once have supported useful responses to infectious challenges associated with pastoralism, changing diets, closer animal contact, and population density. In present environments, parts of the same immunogenetic legacy contribute to autoimmune risk. The finding does not mean that steppe pastoralists carried modern multiple sclerosis in the form diagnosed today, or that everyone with steppe ancestry will develop it. Genetic factors account for only part of the disease, and environment remains important. The evidence shows that a useful immune setting can travel with migrating people, remain unevenly distributed, then acquire a different cost after the disease environment changes. A future population living in a dense coastal city may encounter one set of pressures. Another living in an isolated northern settlement may encounter another. Communities in regions with frequent mosquito-borne disease will continue to face biological conditions unlike those of populations protected by climate-controlled buildings and extensive vector control. People living with broad vaccination coverage, clean water, and rapid access to antibiotics may experience different immune pressures from people whose medical systems are interrupted or unavailable. Global travel brings pathogens together, though it also brings surveillance, vaccines, and treatment. A widespread infection does not automatically create a strong evolutionary change. The infection must affect inherited differences in survival or reproduction, and that effect must continue long enough to alter frequencies. Public health can remove the difference before it accumulates. A persistent local disease can leave a deeper genetic trace than a dramatic pandemic that passes quickly. Migration will complicate every regional path. Human populations are already more connected than most ancient communities, and continued movement can spread variants rapidly across old geographic boundaries. Mixing does not necessarily produce one uniform genome. People do not move randomly, partner randomly, or receive equal access to every environment. Large cities can contain ancestry from many continents while preserving social, economic, dietary, and medical differences between neighborhoods. Small communities can remain genetically or culturally distinct inside a connected nation. Future migration caused by heat, rising seas, conflict, or employment may bring populations into new environments where older adaptations carry different effects. A variant shaped by one history can enter a body now living under another. The result may be useful, neutral, or costly depending on the surrounding genes and conditions. The concept of one future human usually begins with an image: one average height, one face, one skull, one skin tone, one body shape. Ancient DNA replaces that image with overlapping maps. There are maps of ancestry, food, pathogens, fertility, and movement. Their boundaries shift at different speeds. A person can inherit one region of a chromosome through step-related ancestry, another through early farmer ancestry, and another through a lineage that entered much more recently. Selection acts on particular variants inside this mixed genome rather than on the ancestral label as a whole. Even siblings receive different combinations from the same parents. Over thousands of years, recombination breaks old genetic blocks into smaller pieces. Populations may become more mixed while remaining biologically varied at millions of positions. The future body will also differ before inheritance is considered. Bones respond to loading during growth. Jaws respond to chewing. Metabolism responds to nutrition, sleep, activity, and illness. Two children with similar ancestry can develop different bodies when raised under different material conditions. Technology may widen this developmental variation. One child may grow with abundant protein, preventive medicine, low physical loading, and carefully regulated temperature. Another may grow under recurrent infection, food instability, heavy labor, or extreme heat. Their adult bodies can diverge without 10,000 years of genetic evolution. Those developmental differences may then affect health and reproduction, creating new selective conditions for later generations. The evidence, therefore, supports a limited prediction. Human populations will probably remain recognizably one species, with continuing movement and gene flow between them. The record gives no basis for forecasting separate human species within 10,000 years under ordinary levels of contact. It also gives no basis for a single, uniform future body. Different combinations of diet, disease, reproduction, migration, medicine, and development can pull traits in several directions. Some genetic differences may spread widely, others may remain local. Many visible changes may arise anew during each childhood, rather than becoming fixed in DNA. A few traits could follow convergent paths, as adult milk digestion did through different mutations. Most will be influenced by many genes whose effects depend on the environment. This is the first movement away from the central turn and toward the bodies living now. The old pressures were altered by culture, yet they were not replaced by one new pressure shared across Earth. They became a shifting collection of local environments joined by migration and technology. The future will therefore contain many human bodies, each carrying fragments of ancient adaptations inside conditions their ancestors never encountered. The deepest changes may not be visible in the face or skeleton. They may occur among the organisms living in the intestine, where diet, medicine, sanitation, and birth practices can alter a biological community far faster than the human genome itself can move. A dried piece of human waste found inside a rock shelter can preserve a part of the body that no skeleton can show. The material may be 1,000 or 2,000 years old, reduced to a hard mass of plant fibers, mineral particles, human cells, and microbial remains. Researchers call it paleothesis when its human origin can be established with reasonable confidence. DNA recovered from such material does not reveal only what a person ate. It can preserve fragments from the community of bacteria, archaea, viruses, and other microorganisms that once lived inside the intestine. In a study of 8 well-preserved paleofaecal samples from the southwestern United States and Mexico, researchers reconstructed 498 microbial genomes. Among them were 181 genomes that showed strong evidence of originating in the ancient human gut. 61 had not previously been represented by any known microbial genome in the databases used at the time. These samples were limited in number, came from particular dry environments, and were vulnerable to contamination from soil and later handling. They cannot define one universal ancestral microbiome, yet their overall composition carried a clear signal. The ancient samples resembled the gut microbiomes of present-day people living in less industrialized settings more closely than they resembled those of people living in industrialized cities. The human genome had not become a different genome over those 1 or 2 millennia. The ecosystem living inside the body had already moved much farther. A microbiome is not a hidden organ with one stable shape. It is an ecological community whose members compete, cooperate, exchange genes, consume different substances, and respond to the conditions around them. Most of its residents live in the large intestine, where food components that escape digestion by human enzymes become available to microbes. Many plant fibers reach this region largely intact. Bacteria break some of them into smaller molecules, including short-chain fatty acids such as acetate, propionate, and butyrate. Cells lining the colon can use butyrate as an energy source. Microbial products also interact with immune cells, influence the chemical environment of the intestine, and help resist invasion by some pathogens. These functions are distributed across many species rather than assigned to one permanent set of organisms. 2 healthy people can carry noticeably different microbial communities while preserving overlapping metabolic functions. A change in species does not always mean a loss of function, and greater diversity does not automatically mean better health in every circumstance. Still, repeated comparisons across populations show that industrialized microbiomes often contain fewer bacterial species and lack several lineages that remain common in communities with less industrialized lifestyles. The difference appears in a fresh stool sample long before it could appear in bone. One comparison examined people from Matz's hunter-gatherer communities in the Peruvian Amazon, residents of a traditional agricultural community at Tunapuco in the Peruvian Andes, and people living in Norman, Oklahoma. The communities differed in geography, diet, sanitation, medication, animal contact, and many other conditions. Their microbiomes also separated clearly. Some bacterial groups, including members of spirochates, were enriched in the 2 traditional populations and uncommon in the industrialized population. The result did not prove that one community possessed the original human microbiome or that every missing organism was beneficial. Present-day hunter-gatherers and farmers are modern people with their own histories, not preserved copies of the Stone Age. The comparison showed that subsistence and industrialization are associated with distinct intestinal ecosystems, and that microbial lineages, common in one way of life, may become rare under another. A wider African microbiome survey has since made the same warning more concrete. Researchers analyzed communities ranging from rural and horticultural settings to large cities and urban informal settlements. They reconstructed more than 1,000 bacterial genomes that were not adequately represented in existing catalogues. One bacterium, Treponema succinifacans, was widespread in several rural populations and largely absent from urban populations. Its absence was associated with sensitivity to antibiotics, offering one possible mechanism for its disappearance where medication is more common. The study did not reduce every difference to antibiotics. Diet, sanitation, housing, water, parasitic exposure, animal contact, birth practices, and social networks also vary with urbanization. Its most important finding was methodological as well as biological. Much of the world's microbial diversity had remained invisible because microbiome databases were built disproportionately from industrialized populations. A bacterium can appear unusual only because the people who commonly carry it were rarely sampled. This lost visibility matters when researchers compare the present with the past. A sequence recovered from paleofeces may fail to match a named species because that organism has never been sampled from a living population, because its lineage has disappeared, or because ancient DNA is too fragmented for precise identification. The 498 genomes reconstructed from ancient stool therefore provide a partial archive rather than a complete inventory. Even a genuine ancient sample is not a perfect picture of the entire digestive tract. Stool reflects the microbial community of the lower intestine more strongly than the stomach or small intestine. Some organisms disappear after deposition. Others enter from soil. Researchers use human mitochondrial DNA, dietary evidence, preservation patterns, damage signatures, and microbial composition to judge whether a specimen is likely to be human and whether its microbial signal is ancient. The uncertainty remains visible in the language of the evidence. The paleofaces suggest that many industrialized microbiomes have diverged from older configurations. They do not reveal one pure community that every future body ought to recover. The forces producing that divergence can begin at birth. A newborn acquires microbes from the mother, other caregivers, food, skin, household surfaces, animals, water, and the wider environment. Delivery by the vaginal route and delivery by Caesarean section expose infants to different microbial sources during the first hours of life. Although feeding, antibiotics, hospital practices, and later contact can alter the early differences. Breast milk supplies bacteria and complex sugars called human milk oligosaccharides. The infant cannot fully digest these sugars alone. Particular bacteria, including several bifidobacterium species, can use them, helping shape the early intestinal community. Solid food then opens new ecological niches. During roughly the first 3 years, the microbiome develops toward a more adult-like state, though no single sequence applies to every child. A cultural decision surrounding birth, feeding, sanitation, or medication can therefore influence which organisms arrive during a period when the immune system and digestive environment are still developing. This is not genetic inheritance in the usual sense. It is biological transmission through contact and shared life. Some microbial lineages can move repeatedly from parent to child without becoming part of the human chromosomes. The body inherits 2 histories at once: genes passing through egg and sperm, and organisms passing through bodies, households, food, and place. The second history can break much more easily. A human gene does not normally disappear from a family because one person takes an antibiotic. A susceptible bacterial lineage can. Antibiotics are designed to kill bacteria or prevent their growth. They can save life by controlling infection while also disturbing organisms that were not causing the illness. Many communities recover partly after treatment, though recovery varies by drug, dose, age, diet, previous exposures, and the starting microbiome, some species return slowly. Others may remain below detection. Repeated treatment across a population can favor resistant organisms and genes capable of moving between bacterial species. Industrialization, therefore, does not merely remove microbes. It changes the evolutionary conditions acting inside them. A large genomic analysis, published in 2025, found widespread gene-specific selective sweeps across human gut bacterial populations. Industrialized and non-industrialized microbiomes showed evidence of continuing microbial adaptation, although the particular targets and ecological settings differed. The human host may require many generations for a useful genetic variant to rise. A bacterial population can pass through immense numbers of generations within one human lifetime. Its evolutionary response can occur while the host's genome remains almost unchanged. Diet supplies another filter each day. Many industrialized diets contain less microbiota-accessible carbohydrate than diets built around diverse whole plants. A bacterial lineage specialized for degrading a certain complex carbohydrate gains little from living in an intestine where that substrate rarely arrives. Its population may shrink until it becomes difficult to transmit. Experiments in mice carrying a human-derived microbiome have shown that a low-fiber diet can reduce microbial diversity over generations. With some losses becoming difficult to reverse through fiber alone, once particular organisms have disappeared from the community. Reintroducing missing microbes alongside the relevant diet restored more of the lost diversity in that model. Mice are not humans, and a controlled experiment cannot reproduce the social and ecological complexity of industrialization. The experiment demonstrates a plausible mechanism. Food can reduce microbial populations. Interrupted transmission can deepen the loss. And later, dietary improvement may not restore organisms that are no longer present. A future body could therefore receive every nutrient considered necessary for human cells while lacking microbial functions once maintained through continuous transmission. The change would be real even though no human gene had been deleted. Industrial environments also alter contact. Treated drinking water reduces exposure to dangerous pathogens and has saved vast numbers of lives. Sewage systems separate human waste from food and drinking sources. Smaller households, less soil contact, fewer domestic animals, more indoor time, and heavily cleaned surfaces can narrow some routes by which microbes move. These changes should not be described as simple biological mistakes. Many old microbial exposures included parasites, diarrheal pathogens, and infections that killed children. A highly diverse microbiome can coexist with poor health, malnutrition, or heavy pathogen burden. The central question is not whether humans should return to contaminated water or avoid medicine. It is whether protection from harmful organisms also removes neutral or useful companions, and whether future technology can preserve beneficial functions without restoring the dangers that sanitation removed. The answer is still incomplete. Researchers often find associations between altered microbiomes and inflammatory bowel disease, obesity, diabetes, allergy, or other conditions. Association cannot by itself establish whether microbial change caused the illness, resulted from the illness, reflected medication, or followed the diet and lifestyle surrounding it. The same bacterium may behave differently depending on the community around it. A microbial product beneficial at one concentration may become harmful at another. A species missing from one industrialized population may be replaced functionally by another. Claims that one ideal ancestral microbiome would cure modern disease go beyond the evidence. What the record supports is a broad ecological transition. Ancient stool, rural populations, urban populations, and microbial genomes all show that industrialization is associated with changes in composition, diversity, gene content, and transmission. The health meaning of each change must be tested separately. Some losses may reach much deeper than recent urban life. By comparing bacterial genomes from humans and other primates, researchers have identified hundreds of microbial lineages whose evolutionary histories partly followed those of their primate hosts over millions of years. Many appear to have been lost from humans, with faster losses in industrialized populations. This pattern is inferred from microbial family trees rather than watched directly through fossils. It suggests that parts of the gut community accompanied ancestral primates through long spans of evolution, then disappeared when human ecology changed. A microbial lineage can therefore be older than agriculture, older than Homo sapiens, and still vanish within a relatively brief cultural transition. 10,000 more years could carry this process in several opposing directions. Continued industrialization might reduce contact with environmental microbes, standardize diets, expand Caesarean delivery, and expose more people to repeated antibiotics. Under those conditions, some microbial lineages could become rare or disappear from human populations. Global travel could spread other organisms across old boundaries, making distant microbiomes more similar. Climate change could alter food systems, water supplies, parasites, and infectious exposures. Migration could move people into microbial environments, unlike those of their ancestors. Medicine might deliberately rebuild intestinal communities through defined bacterial mixtures, bacteriophages, engineered microbes, or stored samples collected before diversity is lost. A future child could receive selected microorganisms as carefully as a vaccine. Such intervention would not return the intestine to an untouched past. It would create a designed ecosystem whose members were chosen for measurable functions. The body would then be supported by culture at a level earlier humans could not separate from ordinary contact. That possibility brings the story close to its final boundary. The microbiome changes the meaning of the human body because some of its capacities are shared with organisms that are not genetically human. Digestion, immune education, resistance to pathogens, and production of useful molecules emerge from relationships between human tissue and microbial communities. When those communities change, the functioning body can change before the skeleton, face, or human genome shows anything at all. Future humans may remain visually familiar while living with intestinal ecosystems that no person today possesses. Some may carry fewer naturally transmitted species. Others may carry engineered organisms able to process new foods or deliver medicines. Different populations may maintain different microbial libraries, widening biological inequality without creating new human species. The evidence cannot tell us which outcome will occur. It shows that a major part of bodily change can unfold outside the chromosomes used to predict evolution. The final question must now gather every layer together. Inherited genes, development during one lifetime, microbial transmission, and technology that increasingly manages all three. A skeleton buried 10,000 years from now may reveal lighter loading, altered growth, or a changed jaw. Its DNA may preserve variants that rose through reproduction. The most important part of its biology may still be absent from the grave, because the ecosystem that helped keep that person alive disappeared with the soft tissues. To understand what the future will do to the human body, the last evidence must separate the body we inherit from the body we build, repair, and continually redesign. A vial of blood stored inside a modern biobank can hold several different versions of the future. Human DNA extracted from that blood may reveal variants associated with height, metabolism, immune response, fertility, or the risk of particular diseases. The same person's medical record may contain birth weight, childhood growth, medications, blood pressure, operations, pregnancies, diet, occupation, and age at diagnosis. A stool sample may preserve the microbial community living in the intestine. A body scan may reveal the thickness of bone, the distribution of fat, the shape of the airway, or the condition of the joints. None of these records describes the whole person, yet together they show why the human body, 10,000 years from now, cannot be predicted from genes alone. Some features are inherited through egg and sperm. Some are built during childhood by food, movement, illness, stress, and temperature. Some depend on microorganisms acquired from other bodies and from the surrounding environment. Others remain possible only because medicine repairs, replaces, or supports a biological function. The future body will emerge from all of these layers acting together, and each layer moves at a different speed. The slowest clock is inherited genetic change. A variant must already exist or arise through mutation, influence reproductive outcomes under particular conditions, and pass through many generations before it becomes common. The rise of lactase persistence in several pastoral populations shows that this can happen within a few thousand years. Ancient DNA from immune regions shows that pathogens can also shift gene frequencies over comparable spans. Yet the record never shows the entire species changing in one direction. Selection differs across ancestry, diet, disease, migration, and reproductive history. A variant that rises in one region can remain rare elsewhere. A useful effect can disappear when culture changes the environment. Genetic change is therefore real, though it is constrained by inheritance and by the instability of human societies. 10,000 years may contain roughly several hundred generations, depending on average reproductive age. That is enough time for strong and persistent selection to produce a large frequency change. It is not enough to guarantee a new organ, a new body plan, or a single recognizable type of future human. Most traits people notice in a body are influenced by many genes, each with a small effect, interacting with development. Height offers a clear example. Thousands of genetic variants contribute to differences in stature, while childhood nutrition, infection, maternal health, and living conditions can shift average height within only a few generations. A population can become taller without a major genetic transformation when food and health improve. It can become shorter during prolonged nutritional stress, even when inherited potential remains similar. The body visible in adulthood is therefore a meeting between a genetic range and the environment in which growth occurred. The same principle applies to bone density, muscle mass, jaw form, fat distribution, age at puberty, and many features of the immune system. This developmental clock moves faster than inherited evolution. It begins before birth, when maternal nutrition, illness, hormones, placental function, and fetal growth shape organs that will remain through life. It continues during infancy, when feeding and infection influence growth. It becomes especially visible during childhood and adolescence, when bones lengthen, muscles respond to use, teeth erupt, and reproductive systems mature. A future child raised with low physical loading may develop a different skeleton from a genetically similar child raised under repeated movement and carrying. A child eating highly softened food may grow a different jaw from one eating resistant foods that require prolonged chewing. These changes can appear in every generation, even when no relevant allele changes in frequency. They can also disappear when the developmental environment changes. Archaeological bones preserve the results, though they do not always reveal whether the cause was genetic, nutritional, mechanical, or a mixture. This is why a skeleton from 10,000 years ahead might look different without proving that humanity had genetically evolved toward that shape. A third clock moves through biological transmission outside the human chromosomes. The intestinal microbiome begins forming through contact with the mother, caregivers, food, animals, water, surfaces, and other people. Some microbial lineages can pass repeatedly through families and communities. Antibiotics, sanitation, urbanization, diet, Caesarean delivery, and reduced environmental contact can interrupt that transmission. A bacterium may disappear from a population within a much shorter span than a human gene would require to disappear. Another can spread across continents through travel, food systems, hospitals, or deliberate medical treatment. Future bodies may therefore carry microbial functions that are absent today, while losing organisms that accompanied earlier humans for thousands or even millions of years. These changes could affect digestion, immune education, drug metabolism, and resistance to infection without altering one base in the human genome. Researchers are already experimenting with defined microbial communities, bacteriophages that target selected bacteria, and engineered organisms designed to produce useful molecules. The evidence does not show that such methods will become routine or safe across long periods. It shows that culture is beginning to treat an internal ecosystem as something that can be measured and changed deliberately. The fourth clock belongs to medical support. Insulin allows cells to receive a signal that a damaged pancreas may no longer provide adequately. Dialysis replaces some filtering functions of failing kidneys for limited periods. Pacemakers regulate heart rhythm. Cochlear implants convert sound into electrical signals delivered to the auditory nerve. Joint replacements substitute manufactured surfaces for damaged bone and cartilage. Caesarean delivery creates another route through birth. Neonatal intensive care supports lungs, circulation, temperature, and nutrition while an infant continues developing outside the uterus. None of these technologies transforms the human genome merely by being used. They change which bodies can survive, how long they can function, and whether some inherited vulnerabilities continue into later generations. They also change the body directly during one life. A person with an artificial joint walks through a combination of living bone, muscle, nerves, metal, ceramic, or polymer. A person using an implanted device is still biologically human, while part of the body's function now depends on a cultural object placed beneath the skin. Over 10,000 years, the boundary between repair and redesign could become difficult to define. A treatment restores function when it brings a damaged body closer to a familiar range. An enhancement aims to move performance beyond that range. The boundary is not fixed. Eyeglasses correct vision, yet they also allow people to perform tasks that their unaided eyes could not manage. Vaccination trains immune memory before exposure to a dangerous pathogen. Hormone treatment can alter growth, fertility, metabolism, or sexual development. Prosthetic limbs can restore movement, while some designs may eventually perform particular tasks better than biological limbs. Gene therapies already aim to alter cells inside a patient to treat disease, though their availability, durability, and risks vary widely. Most current gene therapies affect somatic cells and are not passed to children. Changes made to eggs, sperm, or early embryos could enter future generations, creating consequences far beyond the treated individual. The technical possibility does not establish that widespread heritable editing will occur. Safety, ethics, regulation, access, and unintended effects remain serious barriers. A gene linked to one desired outcome may influence several others. Editing one variant could reduce one risk while increasing another. Traits such as intelligence, height, longevity, or disease resistance are usually influenced by many genes and environmental conditions, making simple design claims scientifically weak. Still, the existence of heritable editing changes the future question. Natural selection alters gene frequencies through unequal reproduction over generations. Directed editing could change selected sequences before reproduction, potentially moving faster than ordinary selection if societies chose to use it widely. Such use could reduce some severe inherited disorders. It could also create new inequalities between populations with different access or different rules. A variant removed in one region might remain common elsewhere. A trait encouraged by one society might be rejected by another. The result would again be many future bodies rather than one. Reproductive technology may become equally important. In vitro fertilization already allows fertilization outside the body. Eggs, sperm, and embryos can be stored. Pre-implantation testing can identify some chromosomal conditions and particular single gene variants before an embryo is transferred. These methods do not provide reliable control over most complex traits. Their long-term influence depends on how often they are used, which conditions are tested, who has access, and whether the choices alter which variants are passed forward. Artificial gametes created from other cell types remain an area of research rather than an established human reproductive method. If such technology ever became safe and common, it could change age limits, infertility patterns, and the number of people able to contribute genetically to a child. An artificial uterus capable of supporting an entire human pregnancy does not currently exist. Partial external support for extremely premature infants is being investigated in animal models and experimental systems. These uncertainties must remain visible. The future cannot be built from inventions that have not yet been demonstrated. The relevant point is that human reproduction has already moved partly into laboratories and operating rooms, placing new cultural decisions between inherited variation and birth. Climate will act across all four clocks: heat, changing disease ranges, food instability, air pollution, migration, and altered water supplies can affect bodies during life. They may also create persistent selection if inherited differences change reproductive outcomes over many generations. Technology can weaken those pressures through cooling, medicine, protected agriculture, and relocation. It can also distribute protection unevenly. A population living in controlled buildings may experience less direct heat stress than one working outdoors. Children exposed to pollution may develop different lungs even without genetic change. Mosquito-borne disease may enter regions where immune histories differ. Migration can mix variants shaped under older environments while placing people into new developmental and microbial settings. The body 10,000 years ahead will therefore record both planetary change and the unequal systems built to manage it. Life beyond Earth would create an even more unfamiliar mechanical environment, though permanent human settlement away from Earth remains uncertain. Evidence from astronauts shows that reduced gravity can lead to loss of bone and muscle, shifts in fluid distribution, changes in vision, and other physiological effects over months. Exercise and medical monitoring reduce some losses without eliminating all of them. A population living for many generations under reduced gravity could develop differently during childhood, and inherited selection might eventually act if reproduction occurred successfully in that environment. We do not know whether human pregnancy, birth, and development can proceed safely under lunar or Martian gravity. No multi-generational human population has lived away from Earth. Claims about tall, fragile space humans therefore remain speculation. The example is useful only because it reveals how quickly the developmental environment could diverge if culture places bodies somewhere evolution never prepared them to grow. Even on Earth, social differences may produce greater bodily divergence than geography alone. Access to nutritious food, preventive care, clean air, safe work, fertility treatment, antibiotics, and future genetic technologies will not be equal automatically. One population may preserve more biological variation through advanced medicine. Another may continue facing strong mortality from infections that are treatable elsewhere. One group may deliberately maintain microbial diversity, while another loses it through repeated disruption. Some bodies may contain implants, engineered cells, or artificial organs. Others may remain dependent on biological systems very similar to those of people today. These differences would not necessarily create separate species. Gene flow, migration, and reproduction could continue between populations. They could still create visible and invisible biological inequalities inside one species. The future of the human body is therefore partly a question of distribution. Who receives protection, who bears environmental pressure, and which technologies become ordinary enough to shape generations rather than isolated individuals. The archaeological evidence from the last 10,000 years offers a final discipline. Farming did not change every body in the same way. Settlement did not reduce mobility everywhere at once. Softer food did not create perfect uniformity in jaws. Pathogens did not select one immune system for all populations. Medicine did not stop evolution. Each transformation produced a mosaic of responses, delays, reversals, and trade-offs. The next 10,000 years will probably be no cleaner. Some changes may be inherited. Many will arise repeatedly through growth. Others will belong to microbial partners. A growing number may depend on machines, drugs, surgery, or deliberate biological intervention. The future human may look familiar because external anatomy can remain stable while regulation, immunity, fertility, development, and internal ecology change deeply. Another future human may look different because childhood takes place under new nutrition, new loading, or a new climate. Neither body will represent the final form of the species. There is no final form. Evolution has no destination, and technology has no single plan unless human societies create one. The value of this evidence is not that it allows a portrait of a person living 10,000 years from now. It shows where change is already entering the body. It enters through the food placed before a child, the forces placed upon growing bone, the organisms transmitted at birth, the pathogens stopped by a vaccine, the pregnancy supported by medicine, the variant passed through a family, and the technology that changes whether a biological limit remains a limit. A future skeleton may preserve only a small part of that history. The deeper record will lie in relationships between genes, development, microbes, machines, and the social world surrounding them. Human beings once created settlements, herds, stored food, and cities, then discovered that their own bodies had begun responding. We are now creating environments that reach much further inside the body than a field or a wall ever could. 10,000 years from now, will the human body still be adapting to the world around it, or will it be adapting to a world designed by humans themselves?